So, we were talking about DNA repair, and we have already discussed about the photoreactivation repair, then your uh, base excision repair. This base excision repair falls under the category of single strand damage. Now, there is also another mechanism on the basis of this single stranded damage, that how DNA whose one of the strand is damaged by one or two nucleotides or more than that, then how it will be repaired. So out of this, base excision repair plays an important role that you can see here. Just a quick revision about the base excision repair, that most commonly they are repaired by enzyme. First, they are removed by an enzyme called glycosylase. So, DNA glycosylase, they remove the damaged base and make a abasic site. Abasic means a nucleotide without any base. Then finally comes another enzyme called AP endonuclease. AP endonuclease, it actually just like endonuclease do. AP endonuclease creates a nick at the damaged DNA backbone at the AP site. AP refers to apurinic or apyrimidinic site. Finally, the whole of the damaged base has been removed. First, it is initially done by glycosylase. It removes the damaged base, making a apurinic or apyrimidinic site. Then comes the AP endonuclease. It clips the rest of the nucleotide, the sugar, the baseless sugar, and the phosphate attached. Finally, this part will be mended. This part will be mended by DNA polymerase. It will, with their polymerase activity, it will synthesize the new strand using the complementary strand as a template, and the gap will be sealed by the enzyme called DNA ligase. So this was about the base excision repair. We will talk about today about two more another repair mechanisms that could repair the single stranded DNA which is damaged. Not fully, but some of the nucleotides have been damaged. So out of this is your nucleotide excision repair. So what is nucleotide excision repair is, it is saying that nucleotide excision repair, it repairs the damaged DNA which commonly consists of bulky helix distorting damage like the pyrimidine dimerization. I have talked you previously that what is pyrimidine dimer. It means two pyrimidine molecules are getting attached with one another. So suppose this is one pyrimidine, this is another pyrimidine, and they are joined by some covalent bond. Some covalent bond occurs between them to form pyrimidine dimer. Okay, now their original picture I will show you later on in this video. So what it is saying, that it is saying here that in nucleotide excision repair, that it repairs the damaged DNA which commonly consist of bulky helix distorting damage. That means the damaged base is interfering the normal helix structure of the DNA. That will be removed here by the nucleotide excision repair. like your pyrimidine dimers. Best example of pyrimidine dimer is your thymine dimer, right? That is caused by your ultraviolet light. Now in this case of repair, it not only removes that pyrimidine dimer, it not only removes the damage base, but surrounding the damage base about 12 to 24 nucleotide long strands will be removed out. Okay, so in pyrimidine dimerization, or in such type of nucleotide damage which are interfering with the helix structure of the DNA, which are distorting the helix structure of DNA, nucleotide excision repair will mend those things. It will remove that damaged nucleotide, including surrounding the damaged nucleotide 12 to 24 bases. Now these vary among the prokaryotes and the eukaryotes. So damaged regions are removed in 12 to 24 nucleotides long strands in a 3-step process. So that is 3-step. In a 3-step process they will be removed. Okay, so damaged regions are removed in 12 to 24 nucleotide long strands in a 3-step process, which consists of recognition of damage, excision of damage base both upstream and downstream of the damage by endonuclease. So there is special type of endonuclease which will recognize the damage base, and it will cleave. It will not exactly cleave the damage base, just the damage base is present surrounding the damage base in the upstream and in the downstream, they will cleave them. So some normal nucleotides along with the damage base will be removed out, and then again resynthesis of that removed DNA portion will be done. Nucleotide excision repair is a highly evolutionarily conserved repair mechanism, and it is used nearly all eukaryotic and prokaryotic cells. Nearly all eukaryotic cells and prokaryotic cells use this type of repair. Name is a little bit different, but the process is almost the same. In prokaryotes, this NER, this nucleotide excision repair, is mediated by some proteins called UVR proteins. Okay, in the diagram also you will see about the prokaryotes. Okay, I have it has not been mentioned about the eukaryotes by diagram. Just it is saying that the analog of that similarity of that proteins is what type of proteins it will do. Similarity between the nucleotide excision repair between the prokaryotes and eukaryotes will be compared later on. So in prokaryotes, this nucleotide excision repair, in short it is saying NER, it is mediated by UVR proteins, and in eukaryotes many more proteins are involved. In eukaryotes actually XP proteins are involved. XP, from the name given, related to a disorder where mutation in that genes which are causing this nucleotide excision repair, those proteins which are involved in this nucleotide excision repair, if mutation in those genes that will encode this type of proteins for repair will lead to a serious disease called xeroderma pigmentosum. Xeroderma pigmentosum, we will talk about this in later on, and that's why the name comes XP proteins in eukaryotes, because if these proteins get mutated on gene level, then it will lead to a serious skin disorder called xeroderma pigmentosum. Okay, so this was the overview that normally what they do in this nucleotide excision repair, they will remove the nucleotide, the damaged nucleotide which is distorting the helix structure, as well as it will remove some more nucleotides surrounding that lesion, surrounding that damage base, and it is done by some endonuclease enzyme. Okay, and finally that gap, a big gap has been done. That gap will be filled by some type of DNA polymerase and then ligase, right? And it is a very evolutionary conserved repair mechanism. It is used nearly in all eukaryotic and prokaryotic cells. Okay, in prokaryotic cell, this nucleotide excision repair is done by UVR proteins. In eukaryote, it is done by XP proteins, which has not been mentioned here. I will talk to you just now in my notes. Okay, and also the diagram. There is also one more important thing related with this nucleotide excision repair that you will see, and that is your transcription coupled synthesis. Okay, where you will see a strange, not strange, where you will see a mechanism where your RNA polymerase is transcribing the DNA, and suppose that RNA polymerase encounters some lesion which is distorting the structure of DNA. It quickly gets stopped and will recruit this nucleotide excision repair machinery to come and do their work until it will be stalled. This RNA polymers will be stalled, or it will be removed out from that DNA. So this will also, we will see them. Okay, so now I will go to your nucleotide excision repair pathway. Okay, so here it comes. I hope you can able to see it. So what it is saying, nucleotide excision repair works. Nucleotide excision repair works by recognizing the distortions. By recognizing the distortion. Distortion means changes to the structure, to the shape of the double helix. So nucleotide excision repair works by recognizing distortions of the double helical structure of DNA, such as those caused by thymine dimer or by presence of a bulky chemical adduct on the base. Bulky chemical adduct means some chemicals has been interfering with the double helical structure. That also can be detected by your nucleotide excision repair pathway. So such distortions trigger a chain of events. Such distortions will trigger a chain of events. Such distortions of DNA caused by this type of pyrimidine dimers and chemical adducts will trigger a chain of events that will lead to the removal of short single stranded segment that will include that lesion. If you can able to see, let us see. It is saying that such distortions caused by pyrimidine dimer, caused by the chemical adduct, will trigger a chain of events. 3 steps occur in nucleotide excision repair. Okay, and that will lead to the removal of a short single stranded segment that will include your lesion. Lesion means damage base. This removal will create a single stranded gap in the DNA. So it is saying here that due to these distortions in the DNA, it identify a chain of events occur, and it will remove the single strand area, that the area that includes your lesion, and this removal will create a gap in the DNA obviously, and which will be filled by your enzyme called DNA polymerase, using undamaged strand as a template, because your another strand is okay. Another strand is okay. So, and thereby restoring the original nucleotide sequence. So simple thing, if I show you in this diagram, it is a very simple. Let me remove this. So simple thing, suppose two strand is there, five prime, 3 prime, 3 prime, 5 prime. Okay, so in these two strands, suppose the above strand has a lesion, so there is an enzyme called epiendonuclease. What it will do, it will nick out on the upstream and in the downstream, and it will remove that damaged base, including your some normal nucleotides surrounding it. Then comes a type of DNA polymerase which will mend that mistake, and it will repair that mistake by synthesizing new nucleotides by using this undamaged strand as a template. Finally the gap will be filled by your ligase enzyme. And thus you are restoring it to normal. Okay, thus you are restoring it to normal. So in this way your nucleotide excision repair works. Three categories of enzymes are there, but you will see that there is also one more enzyme which will scan the DNA to identify the lesion also. Okay, so let me talk about in detail now. Okay, so first we are actually basically focusing on about the prokaryotic NER, prokaryotic nucleotide excision repair pathway. It is saying that NER in E coli is largely accomplished by 4 proteins. So there are 4 proteins which will perform this work in E. coli, in prokaryotes: UVRA, UVRB, UVRC, UVRD, with DNA polymerase 1 and DNA ligase. So 4 proteins are involved. And what are those 4 proteins? In prokaryotes 4 proteins are involved, right? And these 4 proteins are UVRA, UVRB, UVRC, UVRD. Then comes DNA polymerase 1, and DNA ligase. So these are the 4 proteins. I have missed one more. No, I have said UVR A, B, C, D. These are the 4 proteins, 4 enzymes. Then comes your DNA polymerase 1, which will fill that gap. Okay, and finally the nick will be sealed by your ligase enzyme. Okay, so first what happens? A complex of UVR A and B will scan the DNA with UVRA being responsible for detecting distortions to the helix. I will show you diagram just now. UVRA and UVRB together forms a complex. UVRA and B together forms a complex in the DNA, in a complex, and it will scan the DNA. This complex will scan the DNA to identify any distortion. Especially UVRA actually responsible for detecting distortions to the DNA. Upon encountering a distortion, when they identify that yes, there is distortion in the helix, UVRA will exit from the complex and UVRB then does their function, that is, it will melt the DNA to create a single stranded bubble around the lesion. So first I am talking about UVRA and B. They together forms a complex. It scans the DNA. This complex scans the DNA to identify any lesion, and when they identify the lesion, some distortions in the DNA due to damage base, UVRA then leave the complex from UVRB. UVRB then melt the DNA to create a single stranded bubble around the lesion, just like transcription bubble, okay, because you cannot able to access that wounded strand unless you unwind them, okay. So it creates a temporary bubble around the site of lesion. Next comes UVRC. UVRC will be recruited by your UVRB. UVRB recruits UVRC. UVRC will create it is a endonuclease, right? UVRC is a type of endonuclease. It will create two nicks, two cuts will be done by UVRC. One cut will be done in case of prokaryote. One cut will be done 8 nucleotide away from on the 5 prime side of the lesion. That means your lesion where it is present on the 5 prime side, 8 nucleotide away it will create a nick, and other 4 or 5 nucleotide away the other nick will be done 4 to 5 nucleotide away from the on the 3 prime side of the lesion. So if I draw you, it will be look like this. This is a DNA. I am creating a long one. Okay, it will able to remember, able to observe you in a good way, like it is not working properly. Okay, so suppose there is damage base somewhere here, so this UVRC will creates a nick. It will create a nick 8 nucleotide, 1 2 3 4 5 6 7. We need one more nucleotide, 1 or 2 more nucleotide. Suppose there is more nucleotide, so 8 nucleotide away from the 5 prime site of lesion it will cut. This is your lesion, okay, and another nick will be cut 4 to 5 nucleotide away from the on the 3 prime side, 1 2 3 4, suppose 5 here, okay. So in this way it will create a nick. So UVRC is acting as a, sorry for that, UVRC is acting as a endonuclease. It is acting as a endonuclease, okay. So simple thing, UVRA-B, first they form a complex, it binds to the DNA and scan the DNA to identify lesion. UVRA actually mean responsible for identifying lesion. After recognizing the lesion, then UVRA will leave the complex and UVRB will melt the DNA around the site of the lesion. Then comes UVRC. UVRC creates a nick. Sorry, it creates two nick. One nick will be created on the 5' side of the lesion. Another nick will be created on the 3' side of the lesion. The 5 on the 5' side, where the UVRC will create a nick 8 nucleotides away on the 5' side of the lesion, and on the 3' side it will create a nick 4 or 5 nucleotides away on the 3' side of the lesion. This cleavage, this nick created by UVRC, it will create a 12 to 13 nucleotide long single stranded DNA fragment. This cleavage will create 12 to 13 nucleotide long single stranded DNA fragment that contains your lesion also, which will be accessible by the action of DNA helicase. DNA helicase then fully remove. Okay, normally the job of DNA helicase is to unwind, so it will fully remove that damaged fragment. Okay, and that DNA helicase is actually here called UVRD. So UVRD removes that damaged segment. Finally comes DNA polymerase 1 and ligase fills the resulting gap. So I will now show you the diagram here better. It will be very good to show you the diagram. Okay, here it is. So this is a nucleotide excision repair diagram. In case of prokaryotes, you can see here that there is a distortion shown in orange in the first figure. You can see these distortions will be recognized by this UVRAB complex. They are tetramer, means two subunits of UVRA joined with two subunits of UVRB, and they are all ATP dependent. Okay, because you are moving na, and for movement we require energy. So UVRA and B moves, it scans the DNA to identify any distortion. Upon encountering this distortion, as you can see in the A figure, upon encountering this distortion, you can see here clearly that UVRB is melting the DNA. It has melted the DNA locally around the site of lesion, whereas UVRA will then leave the complex. UVRA leaves the complex. Then comes the figure B. So it melts the DNA around the site of lesion. UVRA leaves the complex, its job is over to identify lesion, its job is over and it goes out. Then UVRB melts the DNA around the site of lesion. See, it has been shown. Then comes your UVRC, which will create a nick on the 5' side and on the 3' side in the lesion. In prokaryotes, UVRC will create a nick on the 5' side, 8 nucleotide away on the 5' side of the lesion. I think they have given accurate information here also. 1 2 3 4 5 6 7 8. Yes, it has been given. After 8 nucleotides, UVRC has created nick on the 5' side, and C on the 3' side. From the lesion, count 1 2 3 4. It has been shown 4. And it has been written also that UVRC will create a nick 4 to 5 nucleotides on the 3' side of the lesion. So on both sides, UVRC creates a nick, thus a fragment of DNA formed, including your lesion, right? And finally, there is another UVR proteins called UVRD. UVRC job is over. It is a type of endonuclease. This UVRC goes out. Okay, now comes the job of UVRD. Then comes then comes your DNA polymerase 1 and ligase. UVRD will remove that fragment containing your lesion, and then the DNA polymerase will actually, what they do, DNA polymerase will fill that gap by catalyzing the synthesis of nucleotides. How? By using the undamaged DNA strand that is your lower end. You can see that in the lower end, in the lower strand, it is undamaged, it is not damaged. So the lower end strand will be recognized as a template for synthesizing the gapped strand. Okay, the gap strand, and thus finally ligase will seal the gap. This is the mechanism normally that is done by nucleotide excision repair pathway. So you can see the diagram clearly. Just for quick recapitulation, you can see, okay, you can see the A figure here, how the UVR A and B scans the DNA and joins to form that complex, and UVR A observe that relation, it then leaves the complex, and after leaving the complex, UVR B melt the DNA. In the figure 8 has been shown that UVR B has melt the DNA, right? But here we can see also that UVRB actually perform the job just after UVRA leaves the complex. It melt the DNA, okay, so that the other proteins can able to access and identify identify that lesion. Then comes UVRC. UVRC acts as an endonuclease. UVRC, what they do, UVRC normally they create a nick. UVRC normally creates a nick, okay. It creates a nick on the 5' side as well as on the 3' side around the lesion, okay. And in case of prokaryotes, it is bit smaller. It creates a nick 8 nucleotides away on the 5' side of the lesion, and it creates a nick on 4 to 5 nucleotides away on the 3' side of lesion. In case of eukaryotes, it will be quite longer, 12 to 24 something nucleotides will be removed, including that lesion. Finally, UVRC job is over. Then comes the helicase UVRD. Helicase called UVRD. It will remove that damaged strand. Okay, that's that fragment that has just been created by the action of UVRC, and it will displace the strand. It creates a gap, and that gap will be fulfilled by your polymerase. Polymerase will fill that gap with ligase, will seal the nick. The last strand, the last nucleotide will be sealed by ligase. So this was about the nucleotide excision repair, okay. Now wait for a minute. Okay, so I have said the basic mechanism of the nucleotide excision repair. Now in case of eukaryotes, it is bit of complicated. It is saying that the principle of nucleotide excision repair in higher cells, that is your eukaryotic cells, is much as same as in E. coli. You have seen in E. coli the repair mechanism in E. coli, the nucleotide excision repair in E. coli and in eukaryotes is almost the same, but the machinery for detecting excising to cleave them, to cut them, and to repair the damage is more complicated. About 25 or more peptides are involved in nucleotide excision repair in eukaryotes. So in eukaryotes, about 25 or more polypeptides are needed for the nucleotide excision repair to complete in eukaryotes. Now in eukaryotes, they use an enzyme, they use XP enzymes, as you can see here, name here XPC. So you will see them, which enzymes are similar with which enzymes in prokaryotes. So it is saying that XPC is responsible for detecting distortions to the helix, a function attributed by UVRA in E. coli. The function of UVRA, that is to identify the lesion that causing distortion in the helix, is same as in case of XPC. XPC also identify the distortion in eukaryotes. Okay, so let me compare it now. So the function of UVRA is similar with XPC in eukaryotes. Okay. Next, as in E. coli, the DNA is opened to create a bubble around the lesion. Obviously, we have to make a single stranded bubble, otherwise how will the other proteins able to cleave the damaged strand? So formation of this bubble involve helicase activities also of the proteins XPA and XPD, which is equivalent to UVRB in E. coli. So the function which was done by UVRB to melt the DNA to form a to melt the DNA to form a single stranded bubble, that will be comparable with your eukaryotic XPA and XPD. So let me write. So UVRB, the function of UVRB is equal to XPA and XPD. Okay, so you are comparing from prokaryotes to with eukaryotes. Okay, the function of UVRB, that is your distortion, it makes a single stranded bubble around the site of lesion, that will be comparable with the function of XPA and XPD. Okay, now the bubble creates cleavage sites on the 5' side of the lesion by the help of a nuclease known as ERCC1 XPF, and on the 3' side by the nuclease enzyme known as XPG, representing the functions of UVRC. So UVRC, as I have shown you in the diagram, that UVRC acts as an endonuclease. This UVRC actually creates a nick on the 5' side as well as on the 3' side. But here in case of eukaryotes, two different enzymes are involved, because eukaryotes are more complicated. They are performing a nick on the lesion on the 5' side by ERCC1 XPF, and on the 3' side it is done by another enzyme called XPG. So let me write it now. So this is UVRC. The function of UVRC is performed by ERCC1 XPF and XPG. ERCC1 XPF creates nick on the 5' side, and ERCC1 XPG performs nick on the 3' side. It creates nick on the 5' side, and XPG creates nick on the 3' side. So this is a comparable features that which proteins in the eukaryotes perform the nucleotide excision repair comparable to the eukaryotes, right? Okay, now in higher cells, the resulting single stranded DNA segment is 24 to 32 nucleotides long, so much high. The fragment that is cleaved by your here XPA and XPD, that same function done by UVRC in case of prokaryotes. So it will displacing the strand, it will displacing the fragment containing the lesion approximately 24 to 32 nucleotides. The gap is finally filled by the action of DNA polymerase and DNA ligase that usually we know. Okay, so this is a comparable feature. I want to show you again so that you can able to identify that which protein in eukaryote perform the nucleotide excision repair. So the function of XPC can be comparable with UVRA to scan the DNA to identify lesion. The function of XPA and XPD can be comparable with UVRB, which will cause a single stranded bubble around the lesion. Then the function of ERCC1 XPF can be comparable with the half function of UVRC. That means ERCC1 XPF will create a nick on the 5' side of the lesion in nucleotide excision repair pathway, whereas XPG will create a nick on the 3' side of the lesion in during this NER pathway, right? Now, and in higher cells the resulting DNA segment is 24 to 32 nucleotides long, and the gap will be finally filled by the action of polymerase and ligase. Okay, now as the name imply, we have named UVR proteins. Why we have named XP proteins in case of prokaryotes, and why we have named UVR proteins in case of eukaryotes in case of nucleotide excision repair pathway enzymes? Why we name them in this way? As the name imply, the UVR proteins are needed to repair the damage from ultraviolet light, because the pyrimidine dimers, how it occurs? By the action of UV light. So that's why we give the scientists have give the name UVR proteins, because they repair the damage that caused by ultraviolet light. The thymine dimer, the thymine cytosine adapt, they are all mended by these UVR proteins. Mutants of these UVR proteins are very sensitive to the UV light and lack the capacity to remove the thymine and thymine cytosine adapt. Suppose the UVR genes, that are creating that are coding for this type of UVR proteins, if these UVR genes get mutated, then it will not able to carry out your nucleotide excision repair process. And these UVR genes are very sensitive to the ultraviolet light also. Okay, so thus if these UVR genes are exposed to the UV light, they will be incapable of carrying out the nucleotide excision repair and lack the capacity to remove the thymine and thymine cytosine dimer. Okay, there is a disease that occurs in humans also called Xeroderma pigmentosum. I think you can able to see the name here, Xeroderma pigmentosum. So this Xeroderma pigmentosum is a serious skin disease caused by mutation in the enzymes, the genes responsible for the nucleotide excision repair pathway enzymes. So suppose I have already said that the enzymes that are responsible for the nucleotide excision repair are also sensitive to UV light, and if there is mutation in these nucleotide excision repair proteins at gene level, then it will not able to carry out your this nucleotide excision repair pathway. Thus human beings will exhibit a serious disease known as Xeroderma pigmentosum. In short, it is called XP. Okay, XP occurs due to mutation in one of the NER genes. NER means nucleotide excision repair genes. Suppose one of the genes getting mutated, then it will lead to use this type of serious disorder, such as genes like XPA, genes that codes for XPA, genes that code for XPC. Okay, if one of them get mutated, then it will not able to mend that mistakes, not mistakes, it will not able to mend that lesions caused by UV light, and that leads to xeroderma pigmentosum. In xeroderma pigmentosum disease, it is affected individuals become highly sensitive to your sunlight and result in skin lesions including skin cancer. They cannot able to go outside in the sunlight because due to the mutation of these XP genes, and thus their skin becomes very sensitive to the sunlight, and it can result in skin lesions, means damage to your skin, resulting in skin cancer too. Okay. Now these nucleotide excision repair pathway are also able to rescue the RNA polymerase in a process called transcription coupled repair. I have said just initially in this video that there is a pathway called transcription coupled repair where the RNA polymerase will further recruit these nucleotide excision repair proteins if there is any distortions or lesions in the DNA. Okay, so what happened in the transcription couple repair? In the transcription couple repair, the RNA polymerase will stall means it will stop after identifying the lesions in the DNA downstream. In the transcription couple repair, your RNA polymerase will stop after identifying the lesion in the DNA downstream. Then RNA polymerase will quickly recruit the nucleotide excision repair proteins to the site of lesion that will either back then that RNA polymerase will either back up, it will wait for the line to become clear, means it wait for that damaged lesion to be mended up, or RNA polymerase will dissociate from the DNA to allow the repair proteins to access to the lesion. Okay, so transcription coupled repair is bit a very mechanical process. Okay, suppose just like that, there is a suppose in a bridge there is some where there is a gap. Okay, so the workers will mend that gap, will mend that damage area, and then allows the traffic to goes on. Just like that, RNA polymerase is first, it is transcribing the DNA. Suppose it identifies the lesion, okay, the damaged strand, it is actually moving on the it is actually moving on a template strand, right? RNA polymers can only synthesize RNA from the template strand of the DNA, right? So suppose on that template strand there is your lesion, so RNA polymers will stop and it will recruit the DNA repair proteins, the nucleotide excision repair proteins, to come into play, and either the RNA polymers will back up, it wait for until the damaged strand will be mended off by the NER proteins, or it will remove out from the DNA and allow the repair proteins to access the lesion. Now the significance of this transcription couple repair is that it focus on the repair enzymes on DNA being actively transcribed. The significance of this transcription couple repair is that it focus on repair enzymes on DNA being actively transcribed. Okay, now there is an enzyme TF2H. I think you have remembered the name during the transcription process. TF2H is the last factor that binds and it acts as a helicase actually. So why this name comes here? Because it is related with the nucleotide excision repair pathway also. TF2H is a helicase enzyme that is responsible for two separate functions. TF2H, it is a helicase enzyme. It is responsible for two different functions. First, TF2H can melt the DNA around the site of lesion during your nucleotide excision repair pathway. TF2H is responsible to melt the DNA around the site of lesion during your NER বা nucleotide excision repair pathway, including your transcription coupled repair also. And second, TF2H can also open the DNA template during the process of transcription. Remember the transcription bubble, as I have said before during the time of transcription lecture, there TF2H is responsible to form that transcription bubble so as to proceed for the elongation process. In nucleotide excision repair also, this TF2H melt the DNA around the site of lesion. That means in prokaryotes it was done by UV or DB. Here TF2H actually do. Okay. Now we have not given the name, right? I have given the name here about XPA and XPD, right? I have shown to you that XPA and XPD mainly responsible for creating the single stranded bubble around the lesion. So why we are naming this TF2H? Because TF2H thus have two subunits. TF2H have two subunits, and that's the name of XPA and XPD. So XPA and XPD actually carries the main melting of the DNA around the site of lesion, and this XPA and XPD are the subunits of TF2H. So you can say directly that TF2H is responsible for making that single stranded bubble around the lesion during nucleotide excision repair pathway, as well as it also melts the DNA during the transcription process to make a transcription bubble. Okay, so that is the significance of TF2H. It is working in transcription as well as it is working on the repair pathway. Okay, so that's the end of your nucleotide excision repair pathway. Now I will talk to you the diagram now. Let us see the diagram of the nucleotide excision repair pathway. I have shown you the diagram before, but I will now show you the diagram of the transcription coupled repair. Simple thing. See the RNA polymerase, it is identifying, it is getting transcribing the DNA to form RNA. The green one is your RNA. Using the sense strand, using the template strand to create that RNA. Suppose RNA polymerase identify the lesion, okay, in the downstream, then RNA polymerase will stall and it will recruit the nucleotide excision repair proteins to come into play, and either the RNA polymerase will back up or it will remove from the DNA, so that your nucleotide excision repair proteins may able to access to that lesion site and able to do their work easily. So this is your transcription coupled repair, as you can see here. Okay, next is your mismatch repair. If I say about the mismatch repair, where it is, let us see. Okay, so this is the mismatch repair. So what it is saying in mismatch repair? Mismatch repair systems are present essentially in all cells to correct errors that are not corrected by proofreading. Mismatch repair is a little bit different. It does not related to the lesions. Mismatch repair is related to the mismatches that may occur during the replication process, right? DNA polymerase have proofreading activity. That means suppose it mistakenly place a wrong base, then it will correct that mistake, but still some type of mismatch may occur during replication process, 1 out of 1000 nucleotides maybe. So at that to mend that mistakes, to correct that mistakes, mismatch repair come into play. Mismatch repair systems are present essentially in all cells to correct the errors that are not corrected by proofreading. This system consists of at least two proteins. So in mismatch repair system, it has actually normally three type of proteins are involved if you observe in prokaryotes, but it is saying principally main that it has two type of proteins. One will detect the mismatch and the other will recruit the endonuclease that will cleave that newly synthesized strand close to the region of the damage. Simple thing, just like nucleotide excision repair. The difference between NER and mismatch repair is that NER is done in case of lesion, in case of damage caused by ultraviolet light mainly, but in case of mismatch repair it does not actually remove the lesion. If there is any mismatch that occurs during the time of replication, suppose the DNA polymerase have failed their proofreading activity, have not able perfectly their proofreading activity to correct the mismatch, then this mismatch repair in pathway will come into play. Two types of proteins are involved. One type of protein will detect the mismatch, it will scan the DNA and detect the mismatch, and another type of protein will recruit endonuclease enzymes to come and cleave the newly synthesized strand close to the region of damage. Okay, now in E. coli, the proteins that are involved are mute class of proteins. We name them as mute. So in E. coli, the proteins that are involved is mute class of proteins, that is your mute S, mute L and mute H. These three proteins are involved in the mismatch repair pathway in prokaryotes. Okay, and the function is simple. One type of protein will enzyme will detect the mismatch, and second type of protein will recruit an endonuclease to cleave that mismatch, not perfectly at the mismatch, but it will cleave the strand which are close to the mismatch, which are close to the site of damage. Now why they have said the newly synthesized DNA strand, why it is saying that the, see carefully what is it is written here, one detects the mismatch and the other recruits an endonuclease that cleaves the newly synthesized DNA strand close to the region of damage. Why they have said? Is because mismatch occur in which strand? The strand which is getting replicated, the newly synthesized strand will have that mismatch. The old strand has not mismatch. The old strand is normal. DNA polymerase is synthesizing the newly strand by using the template parental strand as a template. So mismatch may occur in the newly synthesized strand, and these repair enzymes, the mismatch repair enzymes, will identify the newly synthesized DNA first, and it will detect the mismatch on the newly synthesized DNA. Now how do they do that? We will talk in later in this video. Okay, so in E. coli, three types of proteins are involved. One is mute S, mute L and mute S. Remember mute S as S for scan. Mute S actually scans the DNA and identify for the mismatch. Then comes the mute L. Mute L will recruit mute H also. Okay, I will talk to you now, just now. In case of eukaryotes, they don't have this accurate name, but they have the name as MSH. The mute class of proteins which are involved for the mismatch repair pathway. In case of eukaryotes, they will this same process will be carried out by your MSH. MSH means here, sorry for that, I have not said. MSH actually refers to the mute S homologs, means the function done by mute S, that same function will be done by MSH, mute S homologs. Okay, then MLH. MLH means mute L homolog, means similarity with mute L. The function done by mute L will be similar to the function done by MLH. Okay, mute H will be only present in bacteria, which has an alternative proteins in case of eukaryotes. This is followed by removal of damaged region by exonuclease and resynthesis of the DNA of the gap region by polymerase, and next seeding again by the enzyme called ligase. So I will talk to you now one by one in a flow chart. Okay, so let me go to the camera section. Okay, so I am giving a flow chart here just to make it easily, otherwise it will be a very big video. Mutase is a dimer, right? Mutase is a dimer, means 2 subunit of mutase. This mutase will scan the DNA to recognize the mismatch from the distortion they cause in the DNA backbone. It will identify the mismatches in the newly synthesized strand. And how it identify that which strand is newly synthesized, which strand is not newly synthesized, that I will come you to just a second. So mute A scans the DNA to recognize the mismatch, right, from the distortions they cause in the DNA backbone. After detecting the mismatch, a conformational change will occur on the mute S. Suppose the mute S identify the mismatch, then there will be a structural change, a conformational change in the structure of mutase. This complex of mutase and mismatch containing DNA allows or recruits mute L protein to bind. So mutase scans the DNA, identify the mismatch, a conformational change occur in the mutase. So this complex of mutase and the mismatch containing DNA will allow to recruit mute L to bind. Mute L will bind. Mute L will activate mute H. Mute H is a endonuclease. If you can able to see the name, mute H is an endonuclease that will cause incision, that means nick, on one strand of the DNA near the site of the mismatch. Again I repeat, first mute S identify mismatch, it recruits, then after identifying the mismatch there will be a conformational change in the mutase, and mute S with the mismatch containing DNA will now allow mute L to bind. Mute L will now recruit mute H. Mute H is an endonuclease. Mute H will cause incision. It will create a nick. It will create a nick on one strand near the site of the mismatch. Suppose there is mismatch on near to the strand of the mismatch, it will create the nick. Nicking is followed by specific type of helicase enzyme, that is your, in case of prokaryotes it is done by UVRD. As I have said you before in the nucleotide excision repair, UVRD. So nicking is followed by the action of a specific helicase called UVRD and one of the 3 exonuclease. Helicase will unwind the DNA starting from the incision and moves in the direction of the site of the mismatch. Helicase will unwind the DNA starting from the incision, the incision that was made by the mutate, right, close to the site of mismatch. From that point helicase will start to unwind the DNA. The helicase is here UVRD. It will unwind the DNA starting from the incision and moves in the direction of the site of the mismatch, and exonuclease it actually progressively degrade the displaced DNA strand. So from here I am talking about now. So mute H, what they do when mute L recruit mute H is a endonuclease. It will create a nick, it will create a cut close to the site of mismatch, and then nicking is followed by action of helicase UVRD and one of the 3 exonuclease. The nicking done by mute H will also decides on which side the mute H will create a nick, on the 5 prime side or on the 3 prime side. Depending on that, it will recruit the different type of exonuclease and helicase. Okay, so mute H creates a nick near to the site of mismatch. Either on the 5 prime side it creates a nick, or on the 3 prime side, not on the both. In nucleotide excision repair we have seen in UVRC, right? They are creating a nick on both sides, but here mute H not creating nick on both sides, it is creating nick on one of the sides, and depending on that, different types of helicase and exonuclease enzyme will come into play. So mute H will create a nick on one side near to the site of mismatch, then nicking is followed by action of helicase like your UVRD and one of the 3 exonuclease. Helicase will displace that unwind DNA. Helicase will displace that DNA from the starting point of the incision done by mute H and moves in the direction towards the site of the mismatch, and exonuclease will digest the displaced single strand. Okay, finally the gap will be filled by DNA polymerase 3, okay, by using the undamaged strand as a template, and it will be shielded by a ligase. The overall effect is to remove the mismatch and replace it with correctly base paired nucleotide. So let me see you in diagram. Maybe you cannot able to understand the concept. Okay, so this is your mismatch repair pathway. First mute S you can see. Okay, this mutase is scanning the DNA for identifying mismatch. Here you can see the mismatch is given. T is pairing with G. T is pairing with G, which is a mismatch. T always pair with A or G always pair with T. So mute A's first bind to the newly synthesized strand. And how it identify the newly synthesized strand, that which strand is newly synthesized, which strand is not newly synthesized? It's because a process called methylation which occurs in prokaryotes. Normally methylation occurs on the old strand, but the newly synthesized strand is not methylated at that time. So the repair proteins, the mismatch repair proteins, able to recognize that, oh, this strand is not methylated, so this strand must be newly synthesized strand. And the newly synthesized strand also actually contains the mismatch which has just been replicated by the polymerase. Due to lack of this proper proofreading activity, it creates that mismatch which has to be repaired soon. So mute S then scan the DNA and identify the mismatch. Upon identifying the mismatch, there will be a conformational change. As you can see here, ATP, the conformational change occurs in the structure of mute S, and mute S with the mismatch now recruits the mute L to bind. Mute L then recruit mute H to bind one by one after another, and you can see here the figure that mute H is creating a nick actually here, and mute L just actually allowing the mute H to activate and bind. Mute H will create a nick. Here it is shown it is creating a nick on the 5 prime side, but mute H can also create a nick on the 3 prime side. Depending on that, it will recruit the other enzymes which will come to remove that damaged strand and formation of the template, formation of the newly synthesized DNA. So here you can see a gap is created. Okay, a gap is created, and then it will be sealed. Now here the proteins has not been mentioned here, that which proteins come into play. After the nick created by mute H, then your different other proteins will come into play, like your UVRD, which is a helicase, then exonuclease, some exonuclease will come into play to remove the mismatch base, including some normal base. And finally the base will be filled by your DNA polymerase 3 by using the parental DNA strand as a template. Here in gray has been shown the parental DNA strand, while the red one has been shown the newly synthesized strand. So they are recognizing the new synthesis strand by the action of your methylation process that I will talk to you in describing now. Okay, so this is the process of mismatch DPR pathway, as you can see here. Okay, so now I am going to the hemimethylation part. Okay, so the main question arises in your mind is, how does E. coli mismatch repair system know that which of the two mismatched nucleotides to replace? Mismatch means what? A base will be paired with another base and wrongly base. But how does mismatch TPR enzyme know that which of the base has to be removed? Which one is wrongly placed base? Suppose A pair with G here, mismatch base. The strand containing the A has to be removed, or the strand containing the G has to be removed? That's the question. The main question here arises, the mismatch base can identify, how it identifies the parental strand and newly synthesized strand, because mismatch occurs on the newly synthesized strand, not in the parental strand. The parental strand is normal. When from that parental strand a newly synthesized strand is being created by the polymerase due to lack of proofreading activity or due to mistakes in the DNA polymerase proofreading activity, a mismatch base has been incorporated. So the newly synthesized strand has a more has is actually can contain that mismatch. So repair enzymes, the mismatch repair enzymes can able to know that which strand has to be repaired. It's because due to methylation. Let us see. This applies applicable in case of prokaryotes, not eukaryotes. Eukaryotes don't have this clever trick of methylation. Okay, we will talk to you later on. So the answer that E. coli mismatch repair system know that which mismatch base has to be repaired. The answer is that E. coli tags means it creates some sort of chemical signals, not signal, some sort of chemical information to identify them. So the answer is that E. coli tags the parental strand by transient hemimethylation. Hemi means half methylation. Now let us see. There is an enzyme in the E. coli called DAM methylase. DAM methylase, DAM. This DAM methylase will methylate adenine residues on both strands of the sequence 5' GATC 3'. Within the DNA of prokaryotes, there is a sequence called GATC. This GATC is repeated after a certain sequence. GATC sequence is repeated in every 256 base pair. Means after 256 base pair, you will obviously, for surely, you will find this GATC sequence, and within this GATC sequence there is A. This adenine residue is often methylated by this enzyme called DAM methylase. So I think you can able to understood this GATC sequence. Let me talk to you in by writing. Okay. So it is saying that there is a sequence in prokaryotes, that is your 5' GATC 3'. This GATC sequence is repeated after 256 base pair, and within this GATC sequence the adenine is often methylated. The adenine is often methylated. Who methylated? by the enzyme called DAM methylase. Methylation is carried out by an enzyme called DAM methylase. It is done by an enzyme called DAM methylase. It methylates this adenine residue of GATC sequence, and this GATC sequence is repeated after every 256 base pair. Now this clever trick. So this GATC sequence is widely distributed along the entire genome. It occurs once in every 256 base pair, right? Now when DNA replication occurs, the resulting daughter duplex will be hemimethylated. Suppose DNA replication has been occurred, I am showing you, it will able to understand you now, because without writing you cannot able to understand this concept. Okay, so suppose this is your strand, the two parental strand joined by these bonds, hydrogen bonds, right? And they are all methylated at certain position. They are all methylated at certain position. I am showing you by this red just. I will not write. Okay, I am writing CH3. No, it will be not be possible for me to write because it is bit sleepy. So these red ones, suppose these are the methylation sites of the GATC sequence, and it is carried out by DAM methylase. Now suppose this DNA strand will be now replicated. Okay, this DNA strand will be now replicated. So what will happen during replication? What will happen during replication? New strand will be synthesized, right? New strand will be synthesized. Okay, sorry for that, I am going to wrong way. Wait for it, I have to show you in another picture. It is little problematic for me. Okay, wait for a minute. Okay, so this is your two strands, and this is your these two are the parental strands. Okay, these two are the parental strands, 5 prime, 3 prime, 3 prime, 5, sorry for that, 5 prime, and these are the methylation sites. Suppose these are the methylation sites, okay, done by the DAM methylase, and this methylation occurs on adenine residues of GATC sequence, right? Now during replication what happened? During replication the two strands has to be separated, right? The two strands have to be separated, and these are the methylation sites of the two parental strand. And suppose the newly synthesized strand is being formed, right? The newly synthesized strand is being formed. Okay, five prime, 3 prime, sorry for that, 3 prime, 5 prime, 3 prime, 5 prime, 5 prime, 3 prime. So these two are the newly synthesized strand, right? These two are your newly synthesized strand. So as it is a newly synthesized strand, it is still not methylated now. It is still not methylated because diamethylase will identify in future, a little bit later on, this diamethylase will identify the GATC sequence which has to be methylated on their adenine residues, and it will methylate them. But the newly synthesized strand is still not has been methylated. That's why the repair enzymes, suppose there is any not, I will show you in orange. Okay, suppose there is some distortions in the DNA, sorry, suppose there is mismatch in the DNA, then your repair enzyme can able to access them and able to locate that which one strand is newly synthesized and which strand has to be repaired. Okay, so that's the main logic behind this, about the mismatch repair in prokaryotes. So the DNA replication occurs. When DNA replication occurs, the resulting daughter DNA duplex will be hemimethylated, means methylated on the parental strand only, because for a few minutes diamethylase has not methylated the newly synthesized strand, and this will be an opportunity for the repair enzymes to observe where there is mismatch, and they are definitely observe the newly synthesized strand first. So thus the newly synthesized strand is marked because it lacks a methyl group, and hence this strand can be recognized as a strand for repair. Okay, now mut-H proteins binds at such hemimethylated sites. Mut-H, which will acts as a helicase activity, sorry, it acts as an endonuclease activity. It binds to such hemimethylated sites, but its endonuclease activity is normally latent. Okay, normally mut-H can bind to the hemimethylated sites, but its endonuclease activity is normally latent. Latent means it cannot work alone. That means it will work with the help of mut-H and mut-S. So without mut-H, without mut-L, without mut-S, mut-H will not able to work. It can bind to that newly synthesized strand close to the site of mismatch, okay, but it will not carry out their endonuclease activity unless it interacts with mut-L or mut-S and mut-S. So only when mut-L is contacted by mut-L, or only when mut-H is contacted by mut-L and mut-S located at nearby mismatch, then only mut-H become activated and carry out the function in removing the misincorporated nucleotides. So when mut-H is contacted by mut-L and mut-S at certain locations, then on the site of mismatch, then only mut-H will become activated and carry out the function in removing the misincorporated nucleotide. Mut-H then selectively cuts unmethylated strand, because the unmethylated strand is your new strand which contains the mismatch. So only the newly synthesized DNA is in the vicinity of the mismatch is removed and replaced. So I will give you another diagram for this. It will be for easy to you to see. Let us see. I am trying to draw it, that diagram, in step by step. You can see that this is the first figure showing that the sorry, the parental DNA strands are using as a template to synthesize new strand as shown in red, whereas you can see the GATC sequence are observed in the parental strand as well as in the newly synthesized strand, but the newly synthesized strand is not being methylated now by the dimethylates. It will be methylated later on, and that's the opportunity for the mismatch repair enzymes to identify that which strand has to be repaired if a mismatch base has been incorporated to the newly synthesized strand. And thus you can see here mute H can get close to the site of the newly synthesized strand, and it will create a nick close to the site of mismatch as shown here by TG. Okay, but mute-H cannot work alone. They have to work with the help of mute-S as well as mute-L, otherwise it cannot able to work. So this was the thing about the specificity of the mismatch repair enzyme, that how do they identify that which strand has to be repaired. Okay, now wait for a minute. Okay, I have showed this. So methylation is therefore a memory device, right? That will in not a big memory, just a few moments. After that it will be methylated. So methylation is therefore a memory device that will enables E. coli repair system to retrieve the correct sequence from the parental strand if an error has been made during replication. So methylation is actually a memory, a type of memory for the E. coli repair mechanism, for the E. coli repair system enzymes to correct that sequence of the newly synthesized strand. Now different exonucleases are used. Different types of exonucleases are used to remove the single strand DNA between the nick created by mute-H and the mismatch. I have said before that on which side mute-H is creating the nick, depending on that different exonuclease will be recruited. Depending on whether the mute-H is cleaving the DNA on the 5' side or on the 3' side of the misincorporated nucleotide. Suppose if the DNA is cleaved on the 5' side of the mismatch, then exonuclease 7 or REC-J will be used to degrade the displaced DNA in a 5' to 3' direction. Again I repeat, if the DNA cleaved by mute-H on the 5' side of the mismatch, if mute-H cleave the DNA on the 5' side of the mismatch, then exonuclease 7 or REC-J will degrade the displaced strand in a 5' to 3' direction. Now if a nick done by mute-H, if a nick done by mute-H on the 3' side of the mismatch, then the DNA will be removed, displaced, containing the mismatch by exonuclease 1. It has also been shown 6 also, which will degrade the DNA in a 3' to 5' direction. So there is a figure for this, I will show you. It will be easier for you to remember. Okay, this is the figure. So if mute-H, see the left figure, if mute-H creates nick on the 5' side of the mismatch, if mute-H creates nick on the 5' side of the mismatch, then exonuclease 7 or REC-J will remove the strand containing mismatch, and further it will be filled by your DNA polymerase 3 hollow enzyme. If now see at the B figure, at the right figure. If mut H creates a nick on the 3' side of the mismatch, then exonuclease 1 or sometimes 7 also, 6 also, exonuclease 1 or 6, mainly exonuclease 1 will come to play. Exonuclease 1 will remove the strand, will displace the strand, okay, will displace the strand towards the site of mismatch, and it will be then filled by the polymerase 3 and SSB. SSB, you know, single stranded DNA binding protein, which protects the DNA from, which protects the single stranded DNA from getting rewinding also. Okay, so this is the function of mut H. Mut H creates a nick, and it cannot able to create a nick without mut S or mut L. They need these two things. Then mut H activation done by mut L, so it can able to bind to the site, close to the site of mismatch, but it cannot able to create a nick. Mut L, with the help of mut L and mut S, it creates a nick. Okay, and on which side mut H is creating a nick, depending on that, different type of exonuclease will be recruited for the further process to complete. Suppose mut H is creating nick on the 5' side, as you see in the left figure, then exonuclease 7 or rec J will come into play to remove the strand including your mismatch. If mut H creates a nick on the 3' side, then exonuclease 1 will displace that strand containing the mismatch. Finally, the strand will be filled by DNA polymerase 3, holoenzyme and SSB, that is your single stranded DNA binding protein. In this way your in prokaryotes your mismatch repair pathway occurs. Now I have talked lot about the mismatch repair pathway in prokaryotes. Let us see in eukaryotes. In eukaryotes, we are not actually say too much about the mismatch repair in eukaryotes. Okay, I have talked to you this one. Now, in eukaryotes, eukaryotic cells have a multiple mut S-like proteins. Eukaryotic cells have a multiple mut S-like proteins with different specificities, and they are called as mut S homologs or MSH proteins. Eukaryotes have multiple mut S-like proteins with different specificities. They are called as mut S homologs or MSH protein. They also have MLH and PMS called mut H homologs. So if I say you simplicity that the function of mut S, the function of mut S is done by, in eukaryote it is done by MSH, mut S homologs. Then the function of mut L is done, homologs means different types of mut S homologs proteins are there, okay, which carries out the function just like mut S do. Mut L will be similarity to your MLH, mut L homologs, or your PMS. Okay, I have written small s, PMS, right? Okay, so we have just now said that eukaryotes have multiple mut S-like proteins with different specificities. Mut H, in case of eukaryotes, they have multiple mut S-like proteins with different specificities. For example, one is specific for simple mismatches, like another recognizes small insertions or deletions resulting from slippage during DNA replication. Okay, and it is saying that one more information, a medical information, that due to mutation in the genes, for the human homologs of mut S, suppose I have said na mutase homologs. The mutase homologs functions like a mutase proteins. So the if the genes responsible for the mutase homologs is getting mutated, for example MSH2, homologs get mutated, it can leads to a hereditary non-polyposis colorectal cancer. So suppose there is mutation in your mutase homolog gene in eukaryotes, it can cause you a hereditary, means it can pass on to generation, a non-polyposis colorectal cancer, which is a very rare disease, which is a very rare disorder that can occur. Okay, now in case of eukaryotic cell, they don't have this type of clever trick of methylation. So I have just said about the different little bit of proteins that the homologs of mutase or mutate, right? So eukaryotic cells, they don't have this type of hemimethylation or dimethylase that will methylate and can able to identify that which strand is newly synthesized, which strand is old or parental strand. So how does the mismatch repair system in eukaryotes know which of the two strands to be correct? Now that's a big tricky question. It's research going on. It is saying that lagging strand synthesis, as we know, it takes place discontinuously with the formation of Okazaki fragments. You know about DNA replication. There is leading strand synthesis, there is lagging strand synthesis. In lagging strand synthesis, as you know, it takes place discontinuously with the formation of Okazaki fragments that are joined previously synthesized DNA by DNA ligase. Prior to the ligation step, the Okazaki fragments will be separated from the previously synthesized DNA by a nick. You can see that before ligation, if you see in the lagging strand, the Okazaki fragment that occurs, it has not been joined. It will be later be joined by ligase, right? Okay. So this nick can be thought of being equivalent to the nick created in E. coli by mutH on the newly synthesized strand. Again I repeat, if you see carefully to the lagging strand, there discontinuous DNA synthesis occurs, where every time a primase will come and insert a primer and followed by the elongation of the DNA polymerase, and it will not occur continuously. Another, due to the moving of the helicase from 5' to 3' direction, the 3' 5' strand will be lagging behind. So a discontinuous DNA synthesis is going on, and these Okazaki fragments that we are talking about, it is separated from one another by a nick. This nick that occurs between the Okazaki fragments will be later on sealed. Okay, that's okay, but we are talking about repair system. This nick can be thought of, this nick that occurs between the Okazaki fragment can be thought of as equivalent to the nick done in E. coli by mutH of the newly synthesized strand. The human homologs of mutase or MSH can interact with your pCNA, can interact with the sliding clamp of the replisome, which is the pCNA. pCNA is actually proliferating cell nuclear antigen. It was thought of an antigen before, but it is now confirmed that it is actually a component of the sliding complex, the beta sliding clamp, and this clamp allows the DNA polymerase to perfectly bind to the template strand so that it can able to catalyze the synthesis of DNA. It is saying that human homologs of mutase called MSH, it interacts with this pCNA, the sliding clamp, and it will recruit, and thereby this MSH recruit to the site of the mismatch in the newly synthesized strand, in the lagging strand, not in the leading strand, and it can able to repair the mistakes. In case of leading strand, it has not been confirmed that how do they identify the mismatch in the leading strand. Okay, so this was all about the mismatch repair system and the nucleotide excision repair system. Okay, it is saying also that in interaction with the sliding clamp could also recruit the mismatch repair proteins to the 3' growing end of the leading strand. Again I repeat, it is saying, let me draw for you, it will be better on to draw. It is saying in the leading strand, in a leading strand, the leading, if this is the parental strand and if this is the newly synthesized strand, and suppose this is your, no, it will not be able to recognize you here. If this is the sliding clamp, the beta sliding clamp, and suppose this is your DNA polymerase. Okay, it is moving from 5' to 3' direction. Okay, so it is saying here that this is your clamp, this is your sliding clamp. Okay, this is your sliding clamp. Sliding clamp. So this clamp will be interacted by your some proteins of MSH, so that if there is certain mistakes, suppose if there is some mismatches, suppose there is mismatches in the DNA. Okay, suppose there is mismatches in the DNA, then by interaction with the sliding clamp, MSH will recruit the MLH. Okay, and the other enzymes needed for the mismatch repair system. Now in case of lagging strand, as I have said, in case of lagging strand, you know, sorry for that, in case of lagging strand, you know, if this is the parental strand, so the lagging strand is often discontinuous, which will be further sealed by the DNA polymerase, and ligase, and these are all our Okazaki fragments. And it is discontinuous. If I zoom it out, then it will be helpful for you to see. Five prime, 3', 5', 3'. Okay, so these are the Okazaki fragment. So these Okazaki fragments, this nick, the nick that you are seeing here, this nick can be thought of, the nick can be done by the mute H, and the site for repairing. Okay, this was in case of lagging strand. In case of leading strand, it is saying that they are interacting with the your sliding clamp. Okay, so here it is saying, again I repeat, lagging strand synthesis, as we know, it takes place discontinuously. Lagging strand synthesis, as we know, it takes place discontinuously with the formation of Okazaki fragments. Prior to the ligation step, the Okazaki fragments will be ligated further, and this nick can be thought of the nick created by the MSH, this mute H on the newly synthesized strand. And in case of and it is saying also that these mismatched repair protein in eukaryotes interact with the sliding clamp pCNA. This sliding clamp actually allows the DNA polymerase to properly bind to the template strand, so that it can able to catalyze its polymerase activity. Okay, so by interacting with the sliding clamp, it can able to repair the mismatches in the DNA. And it is also saying that in the leading strand, in case of leading strand, there is no such nick because Okazaki fragment does not occur. Here continuous DNA synthesis occur. So here it is saying that maybe that MSH of the mismatch repair proteins, they interact with the sliding clamp, that is your TCNA, the component of sliding clamp, to recruit the mismatch repair proteins to the 3' growing end of the leading strand. So this was the overall about the mismatch repair protein and mismatch repair pathway and the nucleotide excision repair pathway. These are very important pathway that normally comes in exams,